Good morning, everyone, and very warm welcome to our special webinar plenary presentation, which forms part of the Ideal Hub annual forum. So my name's Stella Valenzuela, and I'll be the host of this morning's webinar. Now, first allow me, on behalf of all those present this morning, to pay my respects to the Gadigal people of the Eora Nation. It is from within their ancestral lands that I'm joining today's event. I'd also like to pay respect to the elders, both past and present, acknowledging them as the traditional custodians of knowledge for this land. I'd further like to acknowledge the traditional custodians of the various ancestral lands from which each of you join us today, and to pay respects to those elders past and present. So today's webinar plenary presentation forms part of our annual Ideal Hub Forum activities. I'm the director of the Ideal Hub, which is an ARC industrial transformation research hub comprised of multiple academic and industry partners focused on developing the next generation of diagnostic monitoring and testing devices. Now, before we commence, I have a couple of housekeeping items. Firstly, we aim to have time at the end for questions, so please use the Q&A box at the bottom of your screen to submit your questions. And I'll also alert everyone to the fact that this webinar is being recorded, and we hope to share the presentation via a link on our Ideal Hub website. So it's now my great pleasure to introduce our special invited speaker, Professor Peter Doherty. As most of you will know, Professor Doherty is an immunologist and pathologist who, with Rolf Zickernagel of Switzerland, received the Nobel Prize for Medicine in 1996 for their discovery of how the body's immune system distinguishes virus-infected cells from normal cells. Peter's had a long and distinguished career, leading research from multiple world-renowned research institutes and centres. These include the Wistar Institute, Philadelphia, and the University of Pennsylvania. He headed the Department of Experimental Pathology at the John Curtin School of Medical Research in Canberra, served as chairman of the Department of Immunology at St. Jude Children's Research Hospital in Memphis, Tennessee, and where he still holds the Michael F. Tamer Chair of Biomedical Research. In 2002, he joined the Faculty of Medicine at the University of Melbourne, and from 2014 has been at the Peter Doherty Institute for Infection and Immunity, a joint venture between the university and the Royal Melbourne Hospital. Importantly, Professor Doherty has been and continues to be a constant and clear voice in representing science and the truth, and generously gives his time in support of many causes. This is especially critical given the many challenges we currently face across healthcare, threats of climate change, and the growing influence of fake news. Therefore, the role of scientists in influencing and shaping future ideas has never been greater. So this morning, Professor Doherty will present to us his plenary talk on the long and short of COVID-19. Thank you, Peter, and I present Peter Doherty. Over to you. Thank you.
So good morning, and happy to be here, kind of. So I've been totally immersed in COVID-19 for the last 2 years almost, 19 months anyway. I'm tweeting a lot about it, and Twitter, of course, is a pathway to insanity, and I'm suffering severe intellectual decline as a result. And also, I've been writing a weekly blog for our website, which is called Setting It Straight, about COVID and infection, immunity, and everything to do with this sort of stuff. So some of what I'll be talking about is up there if you've got hours to spare and you're interested in looking at it. They're now up to number 82, so I've done quite a lot of these. I've worked on infection and immunity all my professional career, which now stretches back more than 55 years, and I'm currently the patron of our institute, which means I don't have any responsibilities, I don't have any power, and I do what I'm told. And basically what I've been doing is doing public communication around COVID-19, because I've been on the public stage quite a bit since the Nobel Prize in 1996. and trying to talk to people about science, the importance of science, and about evidence-based reality. I've written a number of books and tried everything I can to get the message to people that reality actually matters. And I think COVID has introduced us to that idea if we don't already have it. Now, I don't seem to be able to change my, oh, here we are. There, got the wrong button. Okay, we've lived with pandemics through human history, and they became much more prominent, of course, once we formed together into larger agricultural communities and then into towns. Epidemic and pandemic infections became enormously important to us. And as you go back and read Thucydides on the Peloponnesian Wars, the plague of Athens, he talks about the plague of Athens and describes that very effectively. Probably typhus, not plague. This is plague. This was the Black Death of the 14th century. The bacterium came into Europe. It's maintained in rats and transmitted by tick fleas, and was killing a half to a third of the population of cities. Now, that's absolutely devastating when you compare what's happened with COVID-19 with that. And that went on for hundreds of years. And this is, of course, in the era before we understood what infectious diseases were. We didn't really understand that until the 19th century with the work of Louis Pasteur and Robert Koch and people like that. We thought they were miasmas, stuff floating around in the atmosphere. And when I read some of the stuff from anti-vaccination people, I think they're still back in the 14th century and clearly want to live there because they don't believe in vaccines or medicine or whatever. And of course, we don't worry so much about the plague anymore. We have perfectly good broad spectrum antibiotics that treat it. To just emphasize how recent it is, though we understood an infectious disease, the plague bacillus was found by Alexander Yeltsin and Kitaro Shibashura, I think it is. And my life overlapped with that of Yersin. He died in 1943, I think, and I was born in 1940. So this is all very, very recent that we understand what infectious diseases are. Now we are constantly threatened by infectious diseases coming out of nature. Some of them just grumble away in countries for ages. Zika virus grumbled away in the Pacific for years before it hit South America, and suddenly we became very aware of it, causing fetal abnormalities and so forth. West Nile virus grumbled away in Africa, and then suddenly about the year 2000, it started to cause major outbreaks in the United States. Killing birds. It's a pathogen that infects birds, kills birds, and it's transmitted from birds to us by mosquitoes, and it grumbles away now in the United States. We have effectively West Nile virus in Australia. It's called Kunjun virus, but it doesn't seem to cause problems. Now, the pandemic viruses we're always terribly concerned about influenza, and we're also very concerned that viruses that can cause catastrophic infectious disease will jump into us from other types of viruses, not just the influenza viruses, which we're very familiar with from many experiences over the decades. And of course, we all know that CoV-1, the original SARS virus, jumped across from bats into civet cats into us back in 2002, 2003. It didn't get out of East Asia, except it got to Toronto. And it got to Toronto the way these viruses get around, in an infected person on an airplane. And that's the big risk for us, of course, infected people on airplanes. And that's what we worry about with all these infections. There was a movie about something like that called Contagion. It's a very good movie. It was called The Thinking Person's Horror Movie. And because it's for thinking people, it didn't do very well at the box office, but you can view it if you want. And it postulates somewhat of a scenario like we're seeing with COVID, except the virus is much, much more lethal. And we're lucky we aren't in that situation. So the viruses we're particularly worried about beyond influenza, which is maintained in nature in aquatic birds and occasionally jumps across into us, and or can from domestic animal species or from domestic chickens, for instance, often does that though, but doesn't do what this coronavirus has done, SARS-CoV-2. It doesn't immediately start to transmit in us. And so far we've been ducking the bullet on this thing largely when it comes to very severe influenza pandemics, like the one we had in 1918, which is thought to have killed 50 million people worldwide in a world population that is about a third the size of that today. Now, so far as we know, CoV-2 has killed about 5 million. Personally, I think we should probably double that, but it's still nowhere near the 50 million. So before the year 2000, we had 2 coronaviruses circulating in humans. These caused common colds and croup. They were known since the 1960s, and the name coronavirus comes from the very first electron microscope pictures that were taken by a lady called June Almeida in London. And she saw this crown-like structure on the virus, which gave the name corona. And so we haven't worried about them terribly because they can be respiratory problems in kids and so forth. They're not major problems for us. But since the year 2000, 5 more human coronaviruses have come across into the human population. That is, up till 2000, there were 2. There are now 6 currently circulating, with 5 new ones coming across since 2000. The original SARS virus, 2 more common cold coronaviruses that came across in 2004, the Middle Eastern respiratory syncytial virus that came across in 2013, and CoV-2 that came across in 2019, 2020. So, and of course, the original SARS virus, but the original SARS virus burnt out. It's no longer circulating. We never made a vaccine. We just control it with public health policy. It killed about 10% of people that it infects. The current coronavirus kills less than 1% if you've got a decent medical system. And the MERS virus, which is still grumbling away in the Middle East, kills about 30%, but it's much less infectious. So these are dangerous viruses and lots out there. So what's happened that's changed since the year 2000? What's happened is prosperity in China, particularly. And China and Asian countries, as part of their cultural mix, have live animal markets where wild animals are brought into the live animal market, and people buy them for human food. They buy them live. And the same problem is with live bird markets. That's why influenza often comes out of Asia. So my personal view is we're at much higher risk from these things because these viruses are in a context where they can potentially transmit to large numbers of people who are now much more prosperous than they were and are getting on passenger planes and are flying all over the place. And that is, of course, how CoV-2 got around the world very quickly by infecting people who travel on planes. These things travel on planes. Now, there are other viruses that we worry about. The henipaviruses like Nipah, which goes from pigs to humans, from bats to pigs to humans, can also go from bats to humans when they contaminate, for instance, fruit or sap that's being drained from trees that the bats eat and humans also tap. Nipah virus can go between people. There's the Hendra virus, which we know of from horses in Queensland. Terrible, terrible disease, killed very, very few people. And then, of course, there are the filoviruses, Marburg and Ebola. Also, we think bat-carried viruses. There's a features to the bat immune system that allow it to carry a lot of relatively persistent virus infections. There's not a lot of money around to study things like bat immune systems. There may be now, but there wasn't in the past. But we really need to understand more about this comparative medicine, how it is that these species maintain these viruses and carry a lot of them. And they carry a lot more coronaviruses than the ones we've dealt with, CoV-2 and CoV-1. And we don't know, of course, whether the common cold ones also come from them. They very likely do. Marburg and Ebola, again, highly lethal infections. We don't worry quite so much about them because they're so lethal. And when a virus kills large numbers of people very dramatically, very quickly, we jump on that so hard that we hold it down. Because CoV-2 wasn't killing enormous numbers of people. It was bad, and it was infecting a lot of people, which we didn't realize early on, infecting a lot of people who had subclinical infections. We didn't hit it fast enough. The thing we should have done, as I mentioned briefly, is shut down the plane travel. Australia did, and that's one of the main reasons we've done so well with this, that we shut down the planes early, the politicians got medical advice, and we kept it out pretty much. Now, the virus is caused, COVID-19 is caused by the virus SARS-CoV-2. COVID-19 is the name of the disease. It's an RNA virus. Viruses can have RNA or DNA genomes. Any virus is just an inert particle, like a piece of dust, only smaller, that has some genetic material encased in a bit of protein, a bit of fat, and with a bit of sugar stuck on, carbohydrate. And it just drifts around. It floats through the air. We can pick it up off surfaces. And when it gets into our nose, it can infect us. It interacts with a gene, with a protein called ACE2, the angiotensin converting enzyme 2, that's on the surface of many cells throughout our bodies. And so the spike protein, the surface protein you can see there in yellow, attaches to the CoV-2. It then cleaves into two fragments, one of which has the receptive binding domain, the RBD for the CoV-2, which is the thing we try to block with vaccines. And the other bit is a fusion protein, which fuses the virus into the surface of our cell and allows the virus to go into the cell. Now, viruses are basically obligate intracellular parasites. They can only multiply within living cells. So when people tell me viruses come from space, I sort of say, well, where are the living cells in space? Because they come from living cells. That's the only way they can multiply. So it's a bat virus that has jumped into us. It may have jumped by an intermediate species. You'll all be aware of the controversy about where the virus came from. Is it created in a lab? Are the bad guys out there? We love bad guys. We love to think there are bad guys out there. We don't like to think that nature's dangerous and we should actually be doing something smart. We'll just continue to be stupid. We'll build more bombs and we'll bomb the bad guys. And so basically, probably not. There's no evidence that it was ever manufactured in a lab. And I believe from Eddie Holmes, who's just won the Prime Minister's Prize, that's kind of ironic, for his work in getting the genome of SARS-CoV-2 out there very quickly. Eddie tells me there's now increasingly strong evidence that this virus has come out through the Wuhan live animal market, through likely some intermediate species. We worried about the pangolin early on. I don't know what the story is there, and I haven't seen this work from Eddie yet, but he says there's a paper in preparation that will come out. So viruses get into cells, they use the cell as a factory, produce more virus particles come out, they infect more cells than us. We get sicker and sicker as a result until the immune system comes in and cuts that back by getting rid of the factory cells that are making the virus and by neutralizing the virus and stopping it from infecting other cells. And we infect other people because this is an infection in our respiratory tract. We're coughing and spluttering it up. It goes into the air, and of course it's in mucus droplets and tiny bits of slime and snot that are what lines our respiratory tract. Enormously important. The science of slime is really important stuff, and that's the way it exists. All the virus doesn't have a brain, it doesn't want anything, but all it needs for evolutionary success is to multiply and transmit, to maintain in nature. So COVID-19 is caused by SARS-CoV-2. Our best protection is vaccine-induced antibodies that bind to that spike protein, to the receptor binding domain, and stop the virus from attaching to that ACE2 molecule. So that's the basis of protection. Those antibodies can also do other things. They can also target other mechanisms onto the virus that will destroy the virus. And we don't measure those, for instance, in virus neutralization tests, the standard assay where we block infection in a cell culture. So the immunity is a bit broader than just the receptor binding domain, but that's the main immunity that we're interested in with CoV-2 as far as antibodies are concerned. So And that shows you the binding. The ACE2 molecule, the virus binding to the ACE2 molecule by the receptor binding domain on the spike S1, and then the virus going into the cell and making this factory cell start to produce. So I've worked on lots of viruses over the years, all sorts of types. Several types of respiratory infections, big DNA viruses like herpes, a lot like influenza. And I've honestly never seen an infection like this. My interest is in immunity and infection and what we call pathogenesis, how viruses cause disease and how the host response can also contribute to ending that disease, but also making the disease worse. And we see that very much in COVID-19. The host response is in some situations making the disease work. I've never seen a virus that's as complex and as nasty as this one. It's really been a surprise. And it took us some time really to catch up with what the nature of this disease is. Early on, we were saying COVID isn't the flu, but it's like the flu. And we were all thinking of it like flu. And the first mistake that was made globally, and the mistake was made by the Chinese and the Americans and the Europeans, not by us. We stopped the planes, they didn't. So if we're going to avoid these pandemics in the future, we have to have complete openness between various countries. We have complete openness about what's happening. We have to strengthen the World Health Organization and make it essential that people report fully and quickly so that we can, firstly, stop the planes flying out of that region, that country. We'll save an enormous amount of money and an enormous number of lives if we can do that. We held it in Australia when we were just dealing with the earlier variants of COVID-2. But the Delta strain, which is now spreading, is much, much more infectious than either of those strains. It just grows better than the other ones. There's nothing very mysterious about it. It's not an immune escape variant. It hasn't been selected by the immune response, which is what happens with flu. Flu is a virus that mutates a tremendous amount. And we select variants all the time that escape from the immune response. We need another vaccination. With this virus, it looks as though we may need booster shots fairly regularly. We don't know how soon yet. We do know that you need one at 6 months after your second shot, especially if you're older. I'm trying to get one now because I'm 81, so my response is not nearly as good as someone at 30 or 40 because our immune response falls off. So we may need booster shots fairly regularly, but that's no big deal. You've heard, I guess, that there can be adverse events, especially with the second dose of the mRNA vaccine, the Pfizer and Moderna type vaccines. There's this cardiac problem of pericarditis, some cardiomyopathy, particularly affecting younger males. All the evidence is, though, and some do go to hospital, just about everyone gets better. I don't think anyone in Australia has died of it. There was a woman died in New Zealand, someone died in America, in Israel. Maybe there are a few deaths, but very, very few. People get better. And the evidence is that the third shot of that vaccine, much less of a problem. And so, and it's, so it looks good from that point of view. We did know from the beginning that it was causing a horrible respiratory disease, and you can see these awful looking, what we call ground glass opacities, that white stuff in your lung. And when the doctors look at this, they say, how is this person breathing? I mean, they don't seem to have any functioning lung. One of the things that they do is to prone people. That's roll them over on their stomach, because then you access parts of the lungs that are not as badly affected and are not as used as much. That's why heavily pregnant women and obese people are greatly at risk. It's extremely hard for them to be prone on their stomach and to have that additional benefit of being in that position. And there's a lot of heavily pregnant women have been seriously affected by this. And we keep trying to get people to vaccinate, especially young women who may become pregnant or who are pregnant. But there's all sorts of total crap out there put out by people who are either ignorant or evil, trying to persuade women not to get vaccinated. It's appalling. And I find it very, very hard to deal with. Now with COVID, unlike flu. Flu is a respiratory infection. It gets into your lungs, it causes major damage to your lungs. When you die from flu, you drown. You drown because of the enormous amount of fluid that's being made in your terminal bronchi, where bronchioles, where the gas exchange occurs between CO2 goes out, oxygen comes in. That gets basically covered with fluid and it can't work. And that's what kills you in flu. And a part of that is due to the host response, producing too much of toxic molecules, trying to get rid of the virus in people who haven't succeeded in doing that, and that flooding the lung. But with this virus, that can also happen in this disease, we think. But with this virus, you've got the additional factor that the virus gets around the body and the blood. With human flu, that's not a prominent feature of what happens. It's very uncommon to find human flu virus in human blood. But this virus gets into the blood. It's what we call viremic or systemic. And once it's in the blood, that's in the main highways of the body. It can go to any organ. It goes to the heart. It goes to the kidneys. We don't know whether it grows in the blood vessel walls. We think it does, but it hasn't been easy to find that, so maybe it doesn't. It certainly gets into the heart and can kill heart cells. It can kill cells in the kidney. We don't know about getting into the brain, though there are substantial brain effects with this infection. And so SARS-CoV-2 is both a local respiratory infection, a viral pneumonia, and a systemic infection that gets around the body and the blood. And that's what makes it such a horrible infection, because what it's doing is that ACE2 molecules everywhere. What it's doing, it's causing blood clots. It's a coagulopathy. So the reason that CoV-2 patients are gasping for breath, other than those horrible opacities in the lung, is that there are microclots in the little blood vessels serving the terminal bronchi. And those microclots are stopping gaseous exchange. And if you can't get oxygen, you're gone. And that's why oxygen is so important. That's why people are going on ECMO machines. An ECMO machine is a machine that basically oxygenates your blood. It's like a heart-lung machine. So I think we've got something like, I forget whether it's 12 or 20 COVID patients currently on ECMO machines in Melbourne. Now, there are not a lot of ECMO machines around, especially in rural Australia. And so that's really an issue. And so that's why, again, people have to get the vaccine to stop this happening. And it can also cause big blood clots, and big blood clots cause heart attacks and strokes. So we're getting people who have strokes, we're getting people who have heart attacks. It's not the normal cause of death in this. But when you read about a young person dying acutely, I'm always suspicious that may be what's happened. And we don't get as many people coming to post-mortem as we used to do in the past. Post-mortem is not nearly as common as it was. And we use other techniques to try and detect things. But sometimes I think things get missed as a consequence. Now, that's the acute phase of COVID-19. That's what can kill you. That's what can put you in ICU. That's why it can put you in a ventilator onto an ECMO machine. And the virus that's circulating at the moment, Delta, is causing a lot more severe disease in unvaccinated young people. 95% of the people in Victoria who are in ICU units are unvaccinated. And the rare ones who are either very old and have a defective immune system, or they've got some other serious comorbidity, like other lung damage that's prior to COVID. So if you end up in that, if you end up in an ICU, you end up on a ventilator, and people are longer in ICUs and on ventilators than with anything else that the doctors have experienced. They're there for weeks, and they're trying to keep them alive, and they do come out of it sometimes. And so basically, you're not going to be the same ever again after that. You're going to have permanent damage. You're going to have lung damage, all sorts of stuff. And that's what I think of as post-COVID, that after being severely infected, hospitalized after that, your life is saved, but your life is probably going to be shorter, and it's probably going to be substantially compromised. But then again, there's another issue with this called long COVID. And we all know about chronic fatigue and all those issues. Well, here for the first time, we've got a situation where we're diagnosing large numbers of people using a very sensitive test, the PCR test. And we've been using it for public health screening. We've been using it to screen people as they come into the country via airplanes. So we've got an enormous amount. We've never done this before with any other infection. So we have an enormous amount of information if we can manage to pull it all together, because our public record systems, one of the, there are great advantages in the Australian federated system and there are great disadvantages. And one of the great disadvantages is pulling information together and really getting it organized doesn't always work as well as it should do. We could have an enormous amount of information out there would be of great value so long as we can pull it together. So we do know, and this took us a while to find out, that a substantial number of people either have a mild to inapparent infection, especially with the early strains of COVID, of SARS-CoV-2, or but then some of them, and we don't know how many, we know up to 30% from a very good study done at University of New South Wales called ADAPT by Tony Kelleher's group at the Kirby Institute. We know as many as 30% can show some signs of brain fog, malaise, sort of muscle weakness, headaches for up to 3 months at 3 months after infection. And we also know a percentage of those go on to get this very, very distressing disease called long COVID, which can be progressive. It causes long-term debility. And it gets worse. Now, we've seen this before after Epstein-Barr virus infection, the big DNA virus that causes infectious mononucleosis, and is actually a persistent infection in just about all of us. Just about all of us are infected with EBV, but a percentage of those people have gone on to this chronic fatigue. So COVID, in one sense, is opening the door to looking at this whole chronic fatigue syndrome. We don't understand it. But maybe now we will start to do so. It can affect even young children. They generally come out of it sooner, but they can be affected by it. And it's a really concerning problem. Very hard to get really good numbers on it. A lot of the data is self-reporting. Now that's great, and it's great that people do that, but you really want a proper clinical diagnosis and so forth before you take that too seriously. So it's really important to get people on long-term systemic trials where people are sampled repeatedly and looked at with the best available technology. The ADAPT trial is one of those. Some recover, but some don't, and some it actually becomes worse. And it may be as many as 1% who are having really serious problems in the long term. The UK has at least 1 million people reporting an experience of long COVID. So a lot to find out here. We don't know whether the virus is persisting. We don't know whether it's an autoimmune type phenomenon. We don't know whether there's brain damage that's doing this. We don't have a lot of information that there's virus in the brain, but there could be some of these cytokines and things that are circulating and so forth. We just don't understand what's happening here yet. We hopefully will, and hopefully we'll get better treatments for it. Now, our protection against viruses, of course, is by the immune system. The immune system, immunity, comes from the word immunus, which means without tax. Categories of Roman soldiers after the wars were described as being in the genio immunium. And immunity is there to limit the tax of infection. Even though we had vaccination back in the 18th century with Jenner, with cowpox virus, and even before that we had variolation, where people were giving children fully virulent smallpox virus, which they generally survived to protect them from smallpox. We didn't really understand anything much about immunity in the latter half of the 19th century. And then, of course, immunity, which is a very, very complex science, has gone on since there. There's two parts of it: the innate immune system, which is all these cytokines and chemokines and toxic molecules that are made very quickly by all sorts of cells to try and limit an infection right at the beginning. If you don't clear the infection by the other part of immunity, that's the adaptive immune response, which is highly virus specific. And gets rid of a pathogen. If that doesn't work because of age, or you're immunocompromised, or you've been treated with cytotoxic drugs or something, then you can get that innate response going on, and it can be over the top. And we get what we can call cytokine storm, which really does us a lot of damage. That's why when people come into hospital with COVID, first thing they're given is they're given some anticoagulant, and they're giving some, if there's antiviral drugs around, they'd be getting those. And they might actually be giving a monoclonal antibody. These are very expensive, but they have some of them. And then if they progress and they're not getting better, they'll be given dexamethasone, the anti-inflammatory, to try and make them feel a bit better. And that helps a lot. And then, of course, all the other support mechanisms are all that's really left. There's also a molecule called interleukin-6. There are very expensive monoclonal antibodies that will block interleukin-6 that drives that inflammatory cascade of these of the innate immune response. And basically, these are used normally to treat rheumatoid arthritis patients. And with COVID, a lot of the stuff that would normally be there to diminish pain and suffering in rheumatoid arthritis patients is being diverted to COVID to save lives. Adaptive immunity, that's the specific immune response we immunize. It's young in the evolutionary terms, only about 400 million years. We find it first in the bony fish. And all aspects of adaptive immunity are mediated by white blood cells, descendants of the white blood cells. Lymphocytes on the main cell, that wasn't discovered till the 1950s, really, by Jim Gowans in England. And the antibodies, the really important antibody-producing cells, are called plasma cells. They were discovered a bit earlier. They tend to sit in human bone marrow, and they pump out antibody molecules. They descend from what we call the B lymphocytes and B cells. And then people discovered the T cells, the ones that I've worked on. Jacques Miller in Melbourne. We always thought Jacques would get a Nobel Prize. He still hasn't. He's won every other prize on the block, but for discovering the role of the thymus and T cells in the 1960s. And the feature of adaptive immunity that we don't have with innate immunity is immune memory. That is, the immune system remembers that we've seen this before and will react more quickly to it again if it comes back. And it may have antibodies that are still circulating in blood, which can block the infection. All aspects of adaptive immunity work by protein-prone interactions. This is a cartoon of an antibody molecule, an IgG molecule. And here's the binding sites that would block that receptor binding domain on the virus. Fc can attach the antibody to various cells, which would then engulf the virus and destroy that antibody-antigen, antibody-virus complex. The antibodies bind to what we call tertiary structures on proteins in the main. That is, when proteins are folded, I mean, proteins are made as a linear sequence. They're folded, they fold, the antibodies tend to recognize that what we call conformational determinants. And that's why we sometimes get to drugs by studying the antigen-antibody interactions structurally, by structural biology. That's the origin of the anti-influenza drugs, for instance, and some of the anti-HIV drugs. Now, as we know, we get serum antibody levels after the vaccination. They can be measured. They progressively fall. Nothing unusual about that. It happens with all infections. And here's actual data from Israel. Here's modeling. That's the vaccine. After virus infection, they don't fall quite as quickly, but that's falling off after vaccination. The Israelis were the first to start giving this third boost. That was modeled actually by Miles Davenport at the University of New South Wales in a very elegant study. He's a modeler, not an epidemiological modeler, but he models immunity and disease. And he modeled that early in the year and pretty much true to what happened with COVID. And of course, one cell makes one antibody. The other, but you get lots of antibodies made to that receptor binding domain in any individual. These are what we call polyclonal responses. And basically, there are lots of different antibodies that all target to that. We make monoclonal antibodies. That was discovered in the 1970s, and how to get a single cell out, make it produce a lot of antibody in the culture system. And now, of course, we do a lot of this by genetics, just by gene transfer into cells that produce lots of protein. And these monoclonal antibodies are the first of the therapeutics that have been there for COVID-19. President Trump, when he got sick at the end of 2020, was given 8 grams of monoclonal antibody. Now, an enormous amount of this stuff, which probably certainly saved him going to hospital, may have saved his life, because he really was getting quite sick. And that neutralizes the virus and stops the infection. Here's one that's been bought by our government. It's a monoclonal made by Glaxosmithkline. It's called Sotrovimab. It's cross-reactive for all the coronaviruses. It was made originally against COVID-1. And it costs, by the time you give it to someone, it's probably costing $2,000 to $3,000 for a course of for a dose, because it has to be given by intravenous infusion, and it costs at least $1,000 just to buy the product, let alone give it. And we all know how high medical costs are. But this stuff will stick around for 42 days in the blood. Its half-life is 42 days. So Sotrovimab, I think we bought something like 30,000 doses of that. We bought another 15,000 doses, I think, of another monoclonal antibody cocktail called, what is it? Ronapreve. Yeah, that is not as cross-reactive, I think, but also very effective at stopping the infection. Antibodies, all our good vaccines work by making lots of antibodies. They stop systemic infections. Something that's in the blood can be stopped readily by antibody, because we can keep high levels of antibody in the blood. After vaccination, we give it here. The immune response goes on here in the armpit. That doesn't generalize around the body. And then after 6 or 7 days, the immune cells start coming out, and they'll seed out to give you the plasma cells and bone marrow and all the rest of it. Then we can keep antibody levels high in the blood for a very long time. Much, much more difficult to keep antibody levels high up here in the nose. And that's why we're getting breakthrough infections in vaccinated people. People are getting infected. They're getting infected up here in the nose. They can feel quite off for a week or so. And really, they can lose their sense of smell and taste the way you do with COVID, and hopefully get it back. But they don't get severe disease, and they get better. And that's because the antibody that's up in the nose, it's like the virus. It's an inert particle. It doesn't move itself around. The virus doesn't move itself around. These are just inert particles that when they come in, they get into nasal mucus, and they have to meet up if the antibody's going to work. Now, it's much more likely, in fact, that the virus will actually meet up with a cell that's carrying the ACE2 molecule, and that cell will be infected, and the infection will start. As soon as you start getting the infection going, you will get more antibody coming in as you get damage up in the nose, and so you'll get possibly a slower or stop the rate of infection of other cells more quickly. And of course, you'll also turn on the T cells, which eliminate the virus-infected factories. But it's just a dating game. And really, if we had a virus vaccine that we gave up the nose, like some inactivated, live attenuated vaccines, one that's used in Europe and North America for influenza called Medimune, for instance, or have been used for a long time in the Soviet Union, we might get more antibody production up here. But it's actually quite hard to keep a lot of antibody production at local site up here and in the lung. It works a lot better in the gut, actually, because there's a lot more tissue around where they can sit. The plasma cells can sit. All immune responses, adaptive immune responses, are the same. You start with a very small number of precursor cells, which when we're vaccinated get dragged into this lymph node here, and then they select the very few cells that can respond, and they divide like crazy. They divide every 6 hours. We get lots of cells made, and after 6 or 7 days, and also they activate, they differentiate to be memory cells, or cells that will give rise to plasma cells, plasmablasts, or to be precursors of T cells or memory T cells. And after the whole thing's over, a lot of the cells that are expanded, we talk about them being clonally expanded during the acute immune response, will die. But some will stick around to be memory cells that are much more quickly recalled. And some of them will also be seeding out new plasma cells into the bone marrow as those plasma cells die out. This looks at re-stimulating the antibody response. This is in people who have been previously infected. This is in naive people who've never seen the virus before. Data from Ali Elabedi at the University of Washington, terrific scientist. And trained with us at St Jude Children's Research Hospital years back. And so, so you get very good antibody responses and antibody-forming cell responses. This is looking at antibody-forming cells as well as at antibody. The other cell we're very interested in is the killer T cells. This is what I study. Here you can see that little green cell is going to kill this cell. That's a virus-infected cell. It comes along and it kills it, and you can see that cell's dead. Now, when the killer T cell has knocked that cell off, you're destroying the factory. You're also destroying all the product that's in the factory. It's like bombing it. So that brings the infection to a close. This is a normal means of closing out an infection. These don't recognize tertiary structure on proteins. They recognize peptides from the virus, little bits of sequence that are bound into the transplantation molecules on the surface of all our cells. Now, I won't go into that in detail. It's very complex. Structure looks like this. And basically, these T cells are targeted onto the surface of cells in order to destroy them. Here you can see after we infect that, say, a mouse with flu virus in the nose, it grows in the respiratory tract, and then we get this immune response. We get this polyclonal, very diverse memory, and then you get increased numbers of memory cells, and you get a boost up with the secondary response. These are cells coming into the lung. These are killer T cells coming into the lung of flu-infected mice. And these are the top right quadrant, you see the killer T cells themselves. They're there by about day 7. This is a young mouse, and there's a lot of them by day 10. If you've already been primed up, they're there. They're not there at the beginning, but they're there about day 5, and you get lots more of them. So you prime up that killer T cell response, but it's not like antibody immediately available. It has to be re-stimulated, and the cells have to come out. It can give you a measure of protection. Here's virus growth in a naive individual. Here's virus growth after secondary challenge. But it's not immediate, and it has to be recalled. But it's part of our immunity against COVID, and we've got some of that coming from the vaccines we're using, and you probably get a lot more of it after prior infection, in fact. There is some evidence that it's protective in flu, but it's, and it's likely true also in COVID, but it's quite hard to get that information. I won't go on about that. It's complicated stuff, and it's not helping. Now, the other thing we've got now is antiviral drugs. They're just starting to come through. We've got molnupiravir, which is coming through from Merck. Our government has ordered 300,000 doses, not yet fully approved anywhere, and it is being produced, I believe, and I expect it'll be approved for use and out there for use pretty soon in the United States, and maybe a bit later after our TGA has looked at it in Australia. Our TGA is looking at it, actually. I think they have progressed it along, and this is a pill. You take a course of 5 pills. There are 300 courses of this have been ordered. And if it's given to you immediately, you test positive, it will stop the infection and you'll get better. The same is true with flu drugs. If you're tested very early and you're given those drugs very early, they work very well. You have to give it right at the beginning, so you could envisage if we had enough of this, it'd be out there in all the doctors' offices, and it would be given straight away. Of course, there won't be quite enough to do that, but it will save some people when we get it. But I don't see us getting it before next year. And there's another drug from Pfizer. It's a different mechanism. Molnupiravir is what we call a nucleoside analog. It causes mutations in the virus that kill it. There's also a protease inhibitor from Pfizer, which looks very good. And I believe we've ordered 500,000 doses of that. So again, that should be here next year, but it's not here yet. And we may wait a while for it, because it'll also be, could also be issues with supply. Even though we have pre-ordered, will there be enough supply where we would be on that list of people to actually get it? So we shouldn't rely on getting these drugs, but it'll be great when we do get them. We should all get the vaccine as soon as we can. I've been involved in public communication on this. I'd already written a book on pandemics way back in 2013, kind of Pandemics for Dummies. Pandemics, what everyone needs to know. It never sold all that well. You know, who wants to read about pandemics when you can read about the Catholic Church, what everyone needs to know, or modern China, whatever you need to know. But it has been translated into Chinese. And then this year I published an Insider's Plague Year about our experience of COVID-19. A lot of it's in those Setting It Straight articles that are on our website, but there's also a lot about the origin of our institute and some other stuff I wrote for various formats during the year. So there I'll leave it, and we can go on to have some questions.
Wonderful. Thank you so much, Professor Doherty, for making it all seem so straightforward and clear in a sense of a very complex area, and covering this disease that we're just starting to really understand more about. Now, there are questions coming through, but I'll kick off first. And I think from the perspective of the Ideal Hub and our interest in developing detection technologies, look, you were mentioning the use of PCR tests, but going forward, could you comment on the role of monitoring and rapid detection technologies in preventing future pandemics and controlling infectious diseases?
Yeah, well, of course, when you get something new, as we saw with COVID-19, once you've got the sequence, you can set up a PCR test. And we got the sequence as a result of Eddie Holmes really pushing the Chinese, and he was a co-worker on this project. He's a fantastic evolutionary biologist, virologist, and you really should have him talk to you if he hasn't done so already. He's just across at University of Sydney. And so as soon as that sequence was published in January the 15th, by the time we got our index case in Melbourne, a man from Wuhan who knew what was happening and was really responsible, called them up and said, look, I think I've got this. They took him in secure transport to the Alfred Hospital, Monash Hospital, and he got better and all the rest of it. But he was diagnosed by the time he arrived, 9 days later, he was diagnosed by PCR. So, you get a new pathogen coming in. As soon as you know the sequence, you can set up a PCR. Now, all the other tests are more cumbersome. And now we've got all these sort of patient-side lateral flow tests. We've got them for antibodies and for virus and so forth. They're all really useful. We haven't used them. And the reason we haven't used them, not because people are being obstructive, it's just that with a very low background of infection, in Australia, it's been much more sensible to use the PCR test because we've been able to turn these things around in Melbourne. I think New South Wales the same. As long as the labs aren't overwhelmed, we turn them around in less than 24 hours and get the message back to people. So that's always going to be the first line of defense, I think, and it's a fantastic defense to have. But then one of the things, what I've done through all this pandemic is our institute has been having 2-3 times a week, 3 times a week initially, and then tailed off to 2 morning strategy meetings where all the heads of the various groups get together and they discuss exactly what they're doing and what's going on. So, and our institute, I think, is unique in Australia in that it has the academic department of microbiology and immunology at Melbourne University, which is a top department. But it's also got in the building the state virus diagnostic labs for viruses and bacteria, World Health Organization Influenza Centre, and the clinical infectious disease specialists that look after the patients at Royal Melbourne Hospital. They're all in the one building, and we all talk to each other, and these group leaders have all been on these calls. So what I've been listening to is the enormous effort that went on in the diagnostic side. I mean, Vidral, our virus reference lab, was the only people who could do the test. So they had an enormous effort in front of them to get those tests out to the private pathology labs, out to the other hospital labs, get them validated, make sure everyone was testing the same thing. So for several months, that's basically what they were doing, an enormous amount of work to get this going. And then the other thing we've been doing is looking at the rapid tests. Initially, there were a lot of really crap lateral flow tests came on the market. And when they were tested, they were found to be pretty useless, actually. And then some really good ones started to emerge. So these are the sort of testing that we envisage using, say, at an airport or at a school. And now they're starting to roll out and they're starting to be commercially available. But still, the benchmark is the PCR. And if anyone tests positive on one of these things, they need to go and get a PCR test immediately. And they still, for the virus, they still involve doing a nasal swab. There's a lot being done trying saliva too. And maybe some of those will work pretty well too. You don't want to sticking nasal swabs up the nose of kids any more than you have to, and all that sort of thing. So there's been an enormous amount of really practical work going on there. And I was, normally I sit off in a research lab. I was totally unaware of all this happening, but fantastic stuff. And now there are some good, well-validated tests, I think, that can be used effectively.
We're currently working on our and getting closer to some clinical trials around the rapid saliva tests as well for COVID. So lots happening there. Now, a question from the audience. So someone's brought up the question around the role of One Health, I think referring to some of your early comments on the transmission from, animal and crossing over. So what's the role of One Health research in preventing pandemics?
Well, One Health research basically is what the Chinese were doing, and what, of course, they've kind of been condemned for. Well, not just the Chinese, other people in Asia were doing very intensively. They were condemned for it because they were going out and isolating these viruses and trying to understand them. I still think that's really important, that we try to find what's out there, and we keep a monitoring on it. And so people like Linfa Wang, who's now in Singapore, was at our big virus lab in Geelong, the big CSIRO lab for many years. But, the changes in CSIRO and stuff, he eventually went to Singapore. Really terrific guy. He's been developing rapid tests, studying the bats, studying the viruses from bats and so forth. Eddie Holmes has been involved in the analysis of what's coming out of bats for years. So we need to keep up that surveillance program. Now, this is, in a sense, it's a disease that starts with One Health because it jumps from bats, probably into another species, then into us. So it's a One Health disease, but it's not a zoonosis. Some people wrongly describe it. A zoonosis is a disease like toxoplasma or Q fever or leptospirosis that goes directly from animals to us. This doesn't. We don't catch it from bats. Maybe one person caught this thing from a bat and then it started to spread, just random chance. We catch it from other people. That's the same with flu. Again, they can jump into us from other species, but once they're in us, they're a human disease and they're spreading between humans. And once they start to spread between humans, if they're still going directly from chickens to humans, which has been the case with some very lethal flu viruses over the last 20 years in China, then they are zoonoses in a real sense. So One Health it's a way of thinking as much as anything else about this, and thinking about the necessity to keep that analytical approach going.
Okay. So just looking through some of the questions here. So someone's asking, how effective are antiviral nasal sprays like Star Farmer's Viralese likely to be as a preventative option for both vaccinated and unvaccinated?
Honestly, my colleagues are not enthusiastic. My medical colleagues, who are the people I look to for all this, I'm not a medic, and they're not enthusiastic, but you probably don't do any harm by spraying them up your nose. But there is potential there, I suspect, for something much more specific. And also there is some effort to develop a live attenuated COVID vaccine, which you could spray up your nose. Now, if you had something you can just give in a spray up the nose, that might overcome a lot of the ejection, because it would seem much more like a natural infection that people who are against vaccination goes. And I should say, we've done fantastically well with vaccination, getting up to 90%. I mean, there are some other countries who've got there. Israel, maybe not as good as that. Portugal did very well. I think Denmark did very well. And so it says there's not a lot of anti-vaxxers out there, but if you've got 10% of the population that's unvaccinated, you've also at the moment got the under 12s non-vaccinated, you've still got a hell of a lot of people who are susceptible. So it is a real concern. But I think something sprays up the nose. Now, the people who really object to, there was a shearer on the TV, and he was probably a very nice man, but he was saying, I won't get vaccinated. I'm not going to let anyone put stuff that I don't know what it is into my body. And he's got massive tattoos. Now, one of the ways we knew that stuff, when you inject it into the skin, goes in the lymph, the drainage system of the body, to the regional lymph nodes, which are basically filter stations in the lymphatics. one of the ways we knew that if you give stuff into here, it goes into the lymph nodes was experiments that were done back in the 1950s, where people injected carbon black soot into mice and into the skin, and then showed it all ended up in here in the regional lymph nodes. And so I looked at this and I thought, well, that's what happens with tattoos. And has anyone ever done this experiment in humans? So I looked up tattoos, and it's a horrific literature. I mean, the tattooing goes in exactly the same way. I thought that it went more superficially, but it doesn't. Evidently, the tattooists give it, and you get some bleeding, so it goes in fairly deep. But it all goes into the regional lymph nodes. So if you've got a blue or a fluorescent or a green tattoo on your arm, you've got blue, fluorescent or green lymph nodes for the rest of your life. That stuff, that particulate material,
lymph nodes,
very diverse in size, sticks up in your lymph nodes. It causes inflammation. It can cause lymphadenopathy. It causes God knows what. You don't know what's in the tattoo ink. And you're giving it just taking this stuff without any problem at all. I mean, as you understand, if you're on Twitter, and as you understand from our own performance, we are all in many ways quite crazy, but there's a lot of really craziness out there.
Yeah. Look, there's quite a few questions come through, and I'm just conscious of time. I'll just take one more. So the Delta strain was a game changer in the management of the pandemic. What's the risk of further lethal strains arising in the future, and how might we better manage those risks?
Yes, that's of course what we're all thinking about. We're all being tremendously optimistic at the moment because the vaccines, they're not specific for Delta, are still handling Delta pretty well. And actually they've handled all the variant strains, Beta, Mu, that have kind of emerged reasonably well. And so we're hoping all we need to do is keep boosting with this. But if we got a real immune escape variant, of course, then we would have to rejig the vaccine. The drug companies are doing this, but if it's a new one, they would have to start from new. It wouldn't take nearly as long as it's taken to test the initial vaccines because it would just be a much more limited clinical trial. You still need some clinical trial though. And so you can make a variant vaccine against a new strain and get it out. But again, you've got this enormous problem of production, which is why we're really trying to get mRNA vaccine industry going here. And it's the threat that worries all of us. But for the moment, we're okay, and we can do okay with simply boosters with the current one. If you did get a breakthrough infection, you'd probably be more solidly immune than you would be just with the vaccine, but we don't have any good data on that yet. That data's been quite slow to come forward. I've been a bit surprised by that. So basically that is our concern, is that it will do what flu does. It's a much bigger virus than flu. It's a big RNA virus, about 3 times as big, 2.5 times as big. Doesn't mutate at anything like the rate that flu does, but we don't know it does mutate. So, and so what we're hoping is if we do get an immune escape variant, that it will pay what we call a fitness cost. So at the moment, I think of Delta compared with Alpha and the viruses that were early on as like Olympic runners. So the initial virus was like a British Olympic 800-meter runner from 1902 or something. So basically, he was a Brit, he was upper-class guy from Cambridge, he probably drank all the night before, and then he went and ran the race the next day, and that was his performance. But whereas a current Olympic runner will be taken from the whole population, and he will be in very strict training and will run a lot faster. So basically, I think that's Alpha and Delta. Delta is just a virus that as it's mutated, it's grown to a higher level, it's grown quicker, and it infects more people as a result. But once you ask Delta to become not a flat race runner, but you ask it to be a hurdler, and the hurdle is the antibody barrier. The question is, can it do that as well? Or will it suffer what's called a fitness cost? And basically, we're hoping that if it hits a severe antibody barrier, we get an mutant which goes around that. It will still grow a lot less, be a lot less pathogenic, cause a lot less disease, and be much less of a problem. That's the analogy with influenza that we're hoping will happen, but we have no guarantee that's the way it will go. There's an enormous amount of randomness. Now, if we're thinking about what we do with new pandemic viruses coming across, firstly, the one thing I mentioned earlier, we need complete openness across the world about this stuff, and we need countries to be very open, stop the planes from flying out. But the other thing we've already got for the coronaviruses, we need these drugs, because as soon as that virus is detected, if you could treat people immediately, give them just a packet of drugs to take over 5 days, just as we do with an antibiotic, you could likely stop this. So we need a reserve of drugs against these potential threats that can be deployed anywhere across the planet very, very quickly. And it's got to be a global thing. And we have to work better globally on this than we've done. I mean, you know, that moron Trump cut funding for the WHO. I mean, the least helpful thing you could possibly do. But basically, we have no confidence that major countries will not be led by idiots.
Lots to think about, and lots of responsibility on each of us to think carefully about the decisions we make, isn't there?
Including how you vote.
Absolutely, absolutely. Look, Professor Doherty, thank you so much again for your time and for taking the time to come and talk to us today. We're very grateful from the Ideal Hub. We thank you very much. And also for those who've tuned in today and would like to listen to the recording, we'll make this available via our website. So thank you very much, and
okay.
Goodbye.
Okay.
Thanks everyone. Bye. Thanks for attending.
Thanks for listening. Bye.